All right. So first of all, what are cell membranes made out of? What are cell membranes made out of? Phospholipid bilayer. And the reason for that, I'll draw this and then like quickly erase it and like as you would kind of assume, draw a giant cell. But if I had a cup of water and I dumped some phospholipids in there, the phosphate heads go in the water and all the tails are out of water. Why is that? Yeah, who's hydrophobic? Yeah, the tails. So what are the tails made out of that's hydrophobic? What? Yeah, triglycerides, lipids. So it's lipids, specifically triglycerides. And why do I draw, if I'm being correct, here I was being lazy, but why, when I'm being correct, do I draw one tail straight and one with like a bend in it? Yeah, one is saturated and one is unsaturated. Which one is unsaturated, the straight or the bent? The bent is unsaturated. So if we think back, it's been a while. This was Unit 2. Grab that. Put it over here. The way that we made a phospholipid was we took a triglyceride. A triglyceride, I had like this glycerol molecule here in gray, and I have 3 fatty acid tails. They do call them fatty acid tails. And so on those, I basically cut off one of them. Yes. All right. So do you have an ultimate review to turn in? Cool. Yeah, just put it in your in folder. And then same thing, Scarlett, if you have an ultimate review to turn in, just make sure you turn in. I've got a stapler back there. So I've got these 3 tails. I basically cut off one of the tails, and I replace it with the circle thing. What's the circle thing up there? Yeah, the head. What's the head made out of? Yeah, a phosphate. And anytime we think of phosphate, what's the next thing that we think about? Yeah, it's got a negative charge. So the phosphate head is polar, and the lipid tails are nonpolar. Anytime somebody says something about lipids, I automatically think of polarity. I'm like, if you're saying blah, 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 this is a lipid, I'm like, okay, that's nonpolar. Maybe that matters. I, of course, also think that lipids can store energy long term. Anytime I see phosphate, I always think of negative charges, and they're polar, things like that. So I cut off one of these tails, and I replace it with a phosphate. And then if I have a bend in one of these tails, it's unsaturated. So you might remember in these big, long hydrocarbon chains like this, carbons always have 4 bonds if they're stable. See ya? They always have 4 bonds if they're stable. So they'll stay straight also. Whenever I put in a double bond, they'll change. Actually, let me not draw it like that yet. Let me see if you guys can figure this one out. All right, so here's one of these fatty acid tails. All right, one of those is going to bend and one isn't, even though they're both unsaturated. If you had to guess, just trying to use pure logic, one of those is going to get a bend and one is not. And it doesn't have to do with the number. I realize that one over there has 5 carbons and this one has 6, so it's not about the number. And it's not about like, hey, it's this many spaces in versus this many spaces in. Something like that. And in fact, I can even do this. All right, so which of those do you think will bend? One bends and one doesn't. The one on the left or the one on the right? I'll give you a second. Talk with somebody around you if you want, and then we'll vote. If there's no double bonds, it should always be straight. To get a double bond, you increase the likelihood that it gets a bend. All right, how many of y'all think the one on the left will bend? How many of y'all think the one on the right will bend? Okay. This is like a school board election. We had like a 35% turnout or something like that. So this one bends. Why does it make sense that that one does? Now that you know why, or now that you know that it does and this one doesn't, which way does this one bend? Up or down? Up. Why? 4 people simultaneously went like this. So this is a bond. What kind of bond is that if it's a solid line? It's a covalent bond. And I didn't draw them all, but I've got hydrogens at the end of these. So right now, do electrons, are they like stable? Are electrons really stable next to like other electrons usually? No. So right now, there's this big open space. These electrons and these electrons and these electrons and these electrons are all like kind of close to each other. They could bend. So they do. So now it's shaped a little bit more like this. And this is probably beyond AP biology, but they call this a cis double bond. This over here is a trans double bond because they're on opposite sides. They're changing sides. They're not both on the same side. They're on different sides. And so that double bond stays. Have you heard of trans fats? This is the tail of a trans fat. So if it's a trans fat, I can advertise it as a double bond. I can advertise it as unsaturated, but it's still straight, so it's still bad for me. And then for reasons that we don't understand, it's actually worse than just regular saturated fats. You're like, well, what was bad about saturated fats? Why am I supposed to be careful about how much saturated fat I eat outside of calories? Yeah. Yeah, it can kind of clog my arteries. Saturated fats, because the tails are all straight, look like this, and so I can pack them all really close together. When I start getting like a bend in a couple of those tails, I can't pack them as close together. And when I can pack stuff close, that's a solid. When it's not as close, that's a liquid. When it's not close at all, that's a gas. So you might be like, well, wouldn't a trans fat look like that? Just like a saturated fat, so shouldn't it just be equally bad to me? Yes, but for some reason, it tends to inflame our arteries more. So saturated fats and trans fats clog arteries equally, but these, for some reason, kind of like cause swelling and inflammation in my arteries too. Almost like if you just had something, like you're walking and there's like a pebble in your shoe or whatever, and you get like a blister. There's just like something like irritating your skin. So that's why cooking with trans fats in fast food restaurants is illegal. And that's why McDonald's French fries don't taste as good as they used to. Now they're just soggy. They used to be crisp on the outside. Okay, so I've got this cell. They also used to be cheap. When I was first teaching, next door at McDonald's, you could get, for $2.99, you could get 2 double cheeseburgers, fries, and a Coke. That was $2.99. 2 double cheeseburgers, fries, and a Coke. And it was $2.99. I could get a Little Caesars pizza on Monday for $2.50. I could get 2 pizzas for $5 on Mondays only. And Little Caesars used to only sell them in 2s. You couldn't buy a single pizza. You could only buy them in pairs. Their boxes were these big, long pieces of cardboard with paper over it. So, like, if you ever left Little Caesars, you always left with, like, 2, 4, 6, 8, 10 pizzas. Anyway, good times. So I got this cell, and I'm looking at some transport stuff. So we'll start over here. I got some of these dots, and I've got some of these dots. These dots cruise on through. These don't. So what do I know about the black dots versus the red dots? Yeah, non-polar things, good. Non-polar things tend to be able to get through phospholipid bilayers easier. So these are non-polar. These are probably pretty non-polar and pretty small. These are either pretty polar or pretty large. As far as the look, the dots look like they're about the same size. So those guys must be polar. What's it called when stuff just moves in like through the membrane like that from high to low concentration? Yeah, diffusion. What kind of diffusion? Because we've actually learned two kinds of diffusion. When we normally just say diffusion, what's like the full name of that? Yeah, simple diffusion. Good. So this right here is simple diffusion. All right, that guy's going to need some help. So what kind of diffusion is helped diffusion? What's that called? Yeah, facilitated. So this guy over here, facilitated diffusion. So this guy over here is going to need help from like maybe a protein channel. There you go. So if something is too big or too polar or a little bit of both or whatever, then we'll usually have to let it in with like a channel. And that's facilitated diffusion because like we said a moment ago, facilitated means to help, and so it's helped by a protein. If I have no help from anybody else, it's just regular old simple diffusion. If I'm in a chemistry class setting and I'm not talking about cells, you're almost always talking about simple diffusion because you don't use like a protein channel in a typical chemistry setting. All right, for some reason, scientists decided that there's another version of facilitated diffusion not called facilitated diffusion that still uses channels. Does anybody remember what that's called? Yeah, good, osmosis. If it's water diffusing in or out through a channel, then they call that osmosis. So osmosis is basically water diffusion. Now, it still needs help. Why is it logical that water needs help getting through? Yeah, so water's polar. So just like this polar thing can't get in, water's polar, so water can't get in either without a channel. And that's because in the middle of that phospholipid bilayer, I've got those lipid tails, and they fear water. They're hydrophobic. So things like water or other polar things can't get through very well. All right. What other modification could I add to this channel? We used it more like at the end of the year. We mentioned it at this point in like Unit 2, but we like used it more later on. Before pumps, what else could we do where it's still a channel? Yeah, I can add a gate onto it. So now I can have a gated channel. And what I was alluding to is kind of like the nervous system where we had like gated sodium ion channels and potassium channels and stuff. So that's what I was referencing. If you're like, what do you mean like later on in the year? So I can also have a gated channel. Number 3, I kind of ran out of room over there, but that's called a channel. I'll kind of put it right there. And then this is a gated channel. So the nice thing about that is I might have a bunch of stuff out here and not much inside, but maybe I don't want to let it in yet. If I've got a channel, as long as there's more outside than inside, it's automatically going to enter. It's like a doorway versus a doorway with a door. So now I can decide to open and close that, and we learned in the nervous system, this is how I decide to consciously think about this or stop thinking about that and think about this instead. So I can think about this class. All right, done. Instead, I'm going to think about this class. So I can selectively decide when to open and close stuff, and that's me deciding when to think and when to think about something else. Okay, so now all of these were types of passive transport. So what's the opposite of passive? Yeah, active. So everything to the left of this, I'll make it that color, is passive. And now, as we start going this way, these will be examples of active stuff. So now, somebody earlier mentioned a pump. So number 5, a pump. What do I know about active things versus passive things? There's a couple things that I know. Can somebody give me one kind of fact? If it's active or if it's passive? Yeah, if it's active transport, it needs energy. So usually that's ATP, or if it's a proton pump, like you said, maybe it's using electron energy. But it has to have some type of energy, and that energy is almost always ATP. So capital E for energy, and usually ATP. Passive, of course, doesn't. What else do I know? Yeah, with passive, we're moving from high to low concentration, and we say that that's with the concentration gradient. With a pump, I can move things from low to high. So we call that against the concentration gradient. But because that doesn't happen naturally, because normally things spread out evenly, that's going against nature, and that's why I need ATP. I'm basically organizing stuff. The entropy laws of thermodynamics says that things should all spread out evenly. Well, I don't want it to. I want all that crap over there. And so the entropy law says everything spreads out evenly unless I input energy. All things that are organized become disorganized unless I input energy. All disorganized stuff stays disorganized unless I input energy. So that's why we have to input energy like ATP. The next one is a lot less likely to show up on the AP Bio test, but it has. Because again, they have to ask questions that students miss. At some point, you have to be able to tell, like, not only who are the 5s and who are the 4s, but like, hey, is there anybody that's going to get like every single thing right? And if you have like 5,000 students get everything right, then you actually don't know who's the best. You just know who's in the top 5,000. So that's why the older you get, the more tests you take, the harder they get, because at some point they have to make sure, like especially in college, if I've got a classroom of like 150 students, I have to somehow know who's number 1, who's number 2, who's number 3, who's number 4. And that means I can't have like 20 people getting a 100% on a test, because then I can't tell who's better. So that's when you start getting into like nobody made 100. There's 150 people in this class, and like the highest grade was a 92 or something like that. That's where that kind of philosophy comes in. So up here, I could kind of merge these together. I can maybe kind of merge together something passive and something active. They still technically label it as active transport. You could argue, I would even argue that it should be called passive, but I'm just a high school teacher. Nobody cares. So on this one, I have something like passively moving out. And let's say I don't care about blue dots. So those things are passively moving out from high to low. But I really like these black dots. And even though I've got quite a few inside and not many outside, I want as many as possible. So I want to bring those in from low to high. So this side is passive, that side is active. Does anybody remember like how these were working together or what this was called? Yeah, co-transport versus counter-transport. Which one is this one, co or counter? Counter-transport. Why do we call this one counter instead of co? They are helping each other, so to speak. They are working as a team. Yeah, what were you saying, Lorenz? That's true. All right, let me that is the thing that's like different, is one is going high to low and one's going low to high. What if I did this though? I still have one going high to low and one going low to high, but they're moving differently. Yeah. Yeah. If they're going the opposite way, we call it counter. If they're moving the same way, we call it co. Now, earlier, some of you guys were saying earlier about like, well, this one's going that way, or this is called counter transport because one's going low to high and one is going high to low. And both of them, one is going high to low and one is going low to high. So it's just overall which way they move. So it is like which way is the flow. So the idea is the energy that this one gives off when it passively moves from high to low is enough energy to power this one to pull this stuff inside. So in the end, as long as I don't give a crap if blue leaves, then I can pull in this stuff. You might remember I had an analogy earlier in the year where I gave an analogy of like an airport, like a circular door, or if you go to like a fancy restaurant, hotel. Sometimes fancy hotels have that. Not my hotels. Hotels I go to suck, but I've seen them. So if I'm like this, hang on. So the first one, I want to bring the black dots inside. So let's say that this room is the cell, and so this is the doorway. Out here's the hallway. The room where you guys are sitting is right here in the cell. So I want these dots to enter. And so I'm the blue dots. If he's already like in the door, like loaded in the door, the circular door, and I go and push this way as I move, I'm the blue dots because I'm wearing a blue shirt. So as I move from high to low, that's like spinning the door, and then that's like pushing this in. Does that make sense? So that'd be like counter transport. And then same idea. I think this is the baby analogy. The co-transport was if I want this to go in, it's kind of like a baby in the circular door, and I push, then it like pushes the baby in like that. I don't know why it had to be a baby. It could just be like another inanimate, helpless object. All right, so that's co or counter transport. So this one is counter because they're going opposite ways, and this one is co-transport. As far as active transport for like moving individual molecules, 2, maybe 3, 4 molecules at a time, that's pretty much what things look like. When we learned about the sodium-potassium pump, it's a pump and it is pushing both of them in like ways against. So it's just sort of like a pump that's moving like 2 things at the same time. So usually pumps and counter-transport and co-transport and stuff like that, they're usually moving like small numbers of molecules at a time. Like the sodium-potassium pump moves like 3 to 2 when it's moving like sodiums and potassiums in and out. All right, cool. So now, what if we want to move like larger quantities of things? So let's say I've got these dots, or I've got this guy, this bacteria. There's supposed to be eyebrows. He almost looks like a spider, multiple eyes. So what is it called if I like bring in mostly dissolved stuff, mostly liquid stuff into a cell, like a bunch of it all at once? Good. Pinocytosis. All right, and then what about if I gobble up something that's more solid, like maybe I'm eating a bacteria? What was that one called? Yeah, phago. So if I'm mainly bringing in liquid, it's pinocytosis, or cytosis. Osis is a state of, or a condition of. Cyte is cell, pino means to drink. Over here, it's phago. Phago means to eat. Phagocytosis. And then there was one other type of like bulk transport where I'm bringing in quite a few things at once. And it was my favorite one. You guys remember what it was? Yeah, receptor-mediated endocytosis. Good. RME. And the way that works is I'll build some receptors for something that I need, and I'll send them up to the top, up to the cell membrane, and then now, because I have these receptors, whatever things that are out here that I want, I'm more likely to grab them. Because like if these triangles happen to bump into the receptors, they're going to grab. If I just sort of like pinch inward and grab whatever's out here, then maybe I get some triangles, maybe I get a lot of them, maybe I don't, but I'm also going to get a lot of other crap. Here, I'm more likely to get a larger percentage of the stuff that I want. Okay, so aren't there like tons of ways to get stuff in and out of cells then? So how do I decide when to use like a pump versus penocytosis versus RME? That's active, that's active, that's active. So it's not an energy thing. I totally understand why I would use a pump versus a channel. A pump, I can go from low to high. A channel, the advantage is I don't have to burn any energy. But what about pump versus penocytosis versus RME? Why would I choose one over the other two? Yeah.
Penocytosis gives you like a bigger bowl, so you don't have to take as long. And then RME is like lactose, so you don't do other stuff.
Yeah. So penocytosis is super fast. So it has super high speed. How accurate is it? Not very. So every other dot that just happens to be out there, it's going to grab those too. So I've also got these other dots, these green ones, whatever. So I might get a really small percentage of what I want, but it's fast. Receptor-mediated endocytosis, it's a little slower. I've got to build receptors, send them up to the front, and so its speed is like, we'll just say like medium, mid-speed. What about a pump? It's bringing in stuff like 1 or 2 or 3 at a time. Isn't that like super slow? So its speed is really, really low. All right, so why would I ever use a pump if it's the slowest? That sounds dumb. Yeah, more accurate. So we'll say accuracy is really, really high. Penocytosis is super fast, but its accuracy is low. Receptor-mediated endocytosis, it's medium. So what do you want? Speed, accuracy, or kind of medium on both? So your body, your cells, will do whatever they need to do. Sometimes I just need to fix this thing fast, and I can be sloppy. Other times I got to be super precise, and it's this. So in my nerve cells, if I start letting in stuff that I shouldn't, I'm going to start screwing up positives and negatives and voltages, so I'm going to be super precise. All right, next up, we start getting into receptors. So here's a receptor. Here's my nucleus. With my DNA wrapped around histones. Has been drawn the same way for years. C for cotton. All right, so I've got this receptor, but I can also have like this receptor. Let me move that down here a little bit. So what's different about these receptors? One's in the membrane and one's not. Yeah, one's membrane bound. One's stuck in the membrane, and the other one is like inside the cell. All right, so what is it that fits? These are receptors, so I'll label them with R's for receptor. What was it that fits a receptor? Like an enzyme fits with a substrate. What is it that fits with a receptor? What's like the general term for something that fits a receptor? Yeah, a ligand or a ligand. I've kind of heard it both ways, depending on if you go to college, like in one state versus another. Some of the ways that words are pronounced are slightly different. If you go to Georgia or something, I'm sure it's ligand or something like that. All right, so I've got this ligand over here. Maybe it's shaped like this, L. And then I got one over here that's more like a rectangle. Like that. All right, what do I know about this ligand compared to this ligand? What is highly likely to be true? The one on the bottom is likely nonpolar. Yeah. So this guy is going to be more nonpolar. Again, I'm sure we can find some random weird exceptions to these things. And that guy is going to be polar. So the nonpolar thing, just like we saw earlier, nonpolar things can slide through the membrane a little bit easier. So this might slide on through the membrane, hit that receptor, and now that receptor's on, and maybe this receptor can go over here and turn on a gene. What was the beginning of a gene called? What sequence was the beginning of a gene called? Yeah, promoter. What was the end of a gene? What was that sequence? Yeah, terminator or termination sequence. And then we also had another kind of on-off switch ahead of the promoter usually. No, because transcription factors usually land near the promoter to help turn it on. So they are helping turn it on. But what's this sequence that functions as like a sequence that's an on-off switch? Yeah, regulatory. Tearing it up today. Just keep that going for like 24 more hours. 20, 25, 26 hours, something like that. All right, so I've got this receptor over here, and it might like cruise on through here, land on the promoter, and turn the gene on. It might be a transcription factor. Or maybe it's an activator, and instead it lands on a regulatory sequence. Or maybe it's an inhibitor, and it lands on a regulatory sequence and like turns a gene off. But a lot of times, for reasons that I've never really read why it is, usually the nonpolar ones, they hit a receptor, and then that receptor, when it does its transduction, it just moves. It doesn't like activate a guy that activates a guy that activates a guy. The protein-based ones, the polar ones, those tend to activate a protein that activates a protein that activates a protein that does something. For whatever reason, the nonpolar ones, typically their transduction doesn't involve like activating guys that activate guys that activate guys. So they kind of just work like that. Okay, this is nonpolar. So let's say that this was a hormone. What would I know about the category of hormones that it's in? If it's nonpolar, what kind of hormone should it probably be? Nonpolar, what kind of hormone is it? Yeah, it's lipid-based. Which lipid? Cholesterol. And so what do we call those hormones that are cholesterol-based? Steroids. That's probably a steroid hormone because it comes from cholesterol, and it probably has ostro or estro in its name. Testosterone, estrogen, progesterone. So those typically work like this. This guy over here, he's polar. And I think I accidentally said it a moment ago. But what do I know about that ligand? It's not a steroid, so what's it made from? What is it? Yeah, protein. So usually the other hormones are protein-based. They might call them amino acid-based. Why does that name also make sense? To say, well, it's an amino acid-based hormone. Why is it like, oh, I can see why that's also correct. Yeah, proteins are made out of amino acids. Yeah. So it's almost like if something was word-based and you said, oh, we could call it letter-based, you're like, well, yeah, because words are made out of letters. So this is a protein. That means it's made out of amino acids. So this polar ligand is probably protein-based, or they might say, again, amino acid-based. And so the way that it works is a little bit different. That receptor and that ligand have to physically fit just like these do. They have to chemically fit. If I answer a question about receptors, I always need to make sure that I mention the receptor and the ligand like fit. It's not just a ligand hits a receptor. It's all about fitting. It's all about shape. It's just like if you had never seen a lock and you had never seen a key, and I was explaining it, the essence is I have to get you to understand the key has a certain shape. That's why it only fits this one specific lock. This key has this other shape. That's why it only fits this specific lock. It's all about shape. So I would need to say that. And then for them, their transduction process has a cascade. We'll call it a transduction cascade. A protein activates another protein, activates another protein. And then eventually, that like does something. It tells the cell to grow, quit growing, whatever. So these are like enzymes that say grow. Or I turn on a gene that makes enzymes that say grow. So there's kind of two different ways to go about it. What else can I add to my picture there? I have reception. I've got transduction, which in this case is a protein activating a protein, activating a protein. What else could I say about this that I can add? Huh? Yeah, I can also talk about amplification. And so now it's like one of these circles activates like 3 of these proteins, and each of those activates like 3 of these proteins, like that, and it keeps on going. So eventually, one ligand translates into more than one enzyme doing something. So that's amplification. And then the third thing is response. So let me go back and show you the diagram that you're more familiar with. A lot of pictures in this group. There you go. So the last thing I need to mention is response. And I don't want to, if I can, if they give me enough information, I don't want to just say, and then the cell responds. Based off of what's happening in the question, what would that response be? So if it was growth hormone, I would then say, and then the cell responds by, growing. Or if it was insulin, I'd say the cell responds by like bringing sugar into the cell. So try to, if you can, they might just give you general stuff anyway. They might just have something like this and you're like, I don't even know what that is. But you could always say, like, for example, if that signal molecule was insulin, then I would suck sugar into the cell. Or if it was growth hormone and that was a protein, then that would like bring sugar, or I'm sorry, make the cell grow or whatever. All right. One type of receptor that is the most common, I think this came up on one of our free responses. Does anybody remember what kind of receptor is the most common? Yeah. Yeah. They call it a G protein receptor. They'll sometimes call it like a G-linked protein receptor. Yeah, so G. So there's my, oops, I put it E for enzyme. That needs to be R for receptor. So here's my receptor. The way a G-linked system worked, a G-linked protein, is we kind of take this and split it into like 2 jobs. So one of these days, you guys will have a job. And you'll probably be good at it. And a lot of the people that you work with will be good at it. But 10% of them will be abysmal. And so there'll be like 3 dudes, and you're like, I can do twice as much work as those 3 dudes combined. And yet they sometimes don't get fired, and they still have a job, and nobody knows why. So let's say you got this receptor. Same thing. This receptor receives, some of you guys might actually work in like a restaurant right now, and you're like, I already know exactly who you're talking about. You're talking about that dude over there, Mitch. Mitch sucks at his job. If any guys know somebody named Mitch, sorry, I don't teach any Mitches, and so that's why I went with that. But I saw some of you guys smile whenever I said, some of you guys work in a restaurant, and you know somebody like that. So you know somebody like that, don't you? Yeah. Oh, I thought you smiled. Okay. No, that's, you're just smiling at the idea of it, not because you're like, I know who you're talking about. All right, so with this one, this receptor kind of has two jobs. It receives the message and it hands it off. It's efficient. G proteins are like two people that between them can perform one job. They do one people's worth of work, but it takes two of them. So here, this guy receives the message. Nearby is this other protein. It's an enzyme. His job is to hand off the message to the inside of the cell. So I receive it from the outside. He has to hand it off to the inside. Here, one guy does it. He receives the message and does the handing off. So the way that it works is like this. Whenever the ligand fits into that receptor, there's like this in-betweener protein. So I guess you could say it's three people doing one person's job. So there's a protein called G. It stands for GTP. And so what will happen is this ligand hits that receptor, this G protein that runs on GTP, that's why they call it a G protein, will slide over and like hit that enzyme. When it hits the enzyme, let's say the enzyme is supposed to like attach to that ligand, or I'm sorry, that substrate. Does that substrate currently as is fit that enzyme? No. But whenever G slides over, this will change shape. Now, will that substrate fit that enzyme? Sure. So that's how it works. So a G-linked protein system is sort of like I take the receiving part and the handing off the message part, and I split it, and I have a middleman. So receptor, G slides over, activates the enzyme, and now this guy can activate a protein that activates a protein that does something, and he can still amplify as he goes. Just like this pathway up here. And for reasons that I haven't looked in a while, and for reasons that aren't completely understood, this is the most common receptor pathway. So there are many receptor pathways, and this one is responsible for, we think, at least 50% of all receptor types in our body. So go figure. And we think that because we're pretty sure that about 50% of all medicines work on these types of receptors. So if it's by chance, then that means 50% of the receptors are this. So, all right. How you feeling on all that crap up there on the board? So I'll hop out of the way. Okay. Any questions you can think of on like membranes themselves, phospholipids, or any of this transport stuff, or any of this this is what they kind of call cellular communication or cell signaling. If you saw in a college class, or like chapter 5's on cell signaling, that's all this like this stuff right here, all these receptors and transduction and amplification, that stuff. Huh? So prokaryotic cells, they have pretty much, I'm trying to think if there's anything that I know that is different. I don't know of any of this missing from prokaryotic cells. Now, it might be that their receptors, they don't use G proteins or something like that. But like they use channels. I know they use pumps. I know they can bring in bulk stuff. I can't think of a reason that they don't have gated channels, but I don't recall seeing one. So now that you ask that, I'd have to kind of look up to see if they don't have gated channels or something. For the AP Bio test, it wouldn't matter, but you guys are always, like, definitely ask questions, even if it's like, hey, I don't think this will be on the AP Bio test. That's, like, part of the reason of this class, but it's not all of the reason of this class. It's, like, a secondary reason. So now that you say that, I can't recall seeing a gated channel in bacteria, but I also can't think of a reason why not. So I'm not sure. But there's not, like, a major difference. It's not like gene regulation in us versus them where they have operons and there's, like, distinct, like, these are big differences, or they don't have introns or something like that. So very good question, but I'm not sure about gated channels. They do have receptors and all that. Yeah. Any other questions? Because next up after this, that's membranes, transport, cell signaling. So next up is cellular respiration and photosynthesis. Anything on this before I erase it? Yeah. Bulk. So like if I'm bringing in, oh, like this part where they're doing, yeah, I'm sorry, amplification. So I don't know. I was always into like music stuff, not like as a musician, but like audio stuff. So do you know what an amplifier is? You may not. They could be square. Okay, so if you play like a electric instrument, like electric guitar or whatever, you have like the amplifier. That's the thing that will like turn up the volume. So an amplifier section it makes something louder with sound. And so to me, calling this amplification makes sense because I'm making the signal bigger and like louder. So amplify means to like make bigger. It's like turning up the volume. Yeah. Other questions? All right, so I'll erase it. Saw some of you guys get pictures. Anybody else before it's gone? Is that me? Okay. You're welcome. Now you're late to the game. That means that one day if you have kids, you're going to be that one that like misses it. Sorry. That seems funny, but it felt mean. Like sometimes you say something, you're like, oh, they don't have that. And other times you're like, oh, I think I was just mean. So that's supposed to be just funny. You'll be a great mother. All right. So what do you guys want first? Photosynthesis or cellular respiration? I'll let you vote. We should be able to get both of them in there. Photosynthesis? Okay, cool. Photosynthesis. Since I've got this in videoed already, we're done. See you. Good luck on the AP Bio test.